The direct link between smell and emotional brain centers is one of the most unique and powerful features of the sensory system, and it explains why odors can influence behavior so quickly and intensely. Curious. Unlike vision or hearing, which are first processed through the thalamus before reaching higher brain regions, olfactory information takes a more direct route. Signals from odor molecules detected in the nose travel to the olfactory bulb and then project straight into structures like the amygdala and hippocampus. These areas are deeply involved in emotional processing and memory, meaning that smells are tightly coupled with how you feel and what you remember. This direct pathway allows smells to bypass many of the filtering and interpretation stages that other senses go through. Thoughtful. As a result, the response to a scent can be immediate and automatic. You might notice a sudden feeling of comfort, alertness, or even discomfort without being able to explain why. That reaction is the brain activating emotional circuits before conscious thought has a chance to analyze the stimulus. In evolutionary terms, this makes sense because smell was one of the earliest systems used to detect food, danger, or social signals, where speed of response was critical. The connection to the amygdala is particularly important because this region evaluates whether a stimulus is threatening or rewarding. When a smell is associated with a past experience, the amygdala helps determine how you should respond. If a certain odor was previously linked to something positive, like a favorite meal or a safe environment, it can trigger a sense of pleasure or relaxation. On the other hand, if the smell is tied to a negative experience, it can evoke discomfort or avoidance almost instantly. At the same time, the hippocampus plays a role in linking smells to specific memories. This is why certain scents can bring back highly detailed recollections, sometimes from many years earlier. These memories are often more vivid and emotionally charged than those triggered by other senses because of the close anatomical and functional relationship between olfaction and memory systems. The brain essentially stores these associations as integrated packages of sensory and emotional information. Another important aspect is that these responses often occur without conscious awareness. You may not actively notice a smell, but it can still influence your mood, decisions, and behavior. For example, subtle environmental scents can affect how comfortable you feel in a space or how you perceive other people. This unconscious processing makes olfaction a powerful driver of behavior because it operates continuously in the background. The strength of these associations can also increase over time through repeated exposure. When a particular smell is consistently paired with a certain experience, the neural connections linking them become stronger. This is a form of associative learning, where the brain becomes more efficient at predicting outcomes based on sensory input. As a result, even a faint hint of a familiar odor can be enough to trigger a full emotional or behavioral response. Because of this direct and efficient wiring, smell has a unique ability to shape both immediate reactions and long-term patterns of behavior. It influences how environments are perceived, how memories are stored and recalled, and how emotional states are regulated, all through pathways that are deeply embedded in the brain's core processing systems. The integration of smell and taste in shaping the perception of flavor is a central mechanism through which the brain evaluates what you consume and decides how to respond. Although people often think of taste as something that happens only on the tongue, the reality is that flavor is a combined sensory experience involving both gustatory and olfactory systems. Taste receptors on the tongue can detect basic categories such as sweet, salty, sour, bitter, and umami, but the richness and complexity of what you perceive as flavor largely comes from smell. When you eat, volatile compounds from food travel up through the back of the nasal cavity in a process called retronasal olfaction. These compounds activate olfactory receptors, sending signals to the brain that are integrated with taste information. This combined input is processed in areas like the orbitofrontal cortex, where the brain assigns value to the experience. This value is not just about identifying the food, but also about determining whether it is rewarding, safe, or worth seeking out again. This integration has a strong influence on behavior because it directly affects motivation and preference. If a particular combination of taste and smell is associated with a positive outcome, such as energy intake or pleasure, the brain reinforces that association. Over time, this leads to increased desire for those foods. Conversely, if a flavor is linked to a negative experience, such as illness or discomfort, the brain can create a lasting aversion. This process can occur rapidly, sometimes after just one exposure. highlighting how tightly linked these sensory systems are to survival mechanisms. The interaction between smell and taste also explains why food can seem bland when olfactory input is reduced, such as during a cold or nasal congestion. In these situations, the taste receptors are still functioning, but without the contribution of smell, the brain receives incomplete information. This demonstrates how dependent flavor perception is on the integration of both systems rather than either one operating alone. Another important factor is that this sensory integration is highly adaptable. The brain continuously updates its interpretation of flavor based on context, experience, and internal state. For example, hunger can enhance the perceived reward value of certain flavors, making them more appealing. Similarly, repeated exposure to specific foods can alter how they are perceived, sometimes increasing preference, even if the initial reaction was neutral or slightly negative. This adaptability is driven by neural plasticity in the circuits that process taste and smell. The more frequently certain combinations of sensory inputs are experienced, the stronger the associated neural pathways become. This not only influences immediate perception, but also shapes long-term dietary habits. Cultural and environmental factors play a role here as well, as repeated exposure to specific flavors within a given context can condition preferences that persist over time. The integration process also involves expectations and prior knowledge. Visual cues, texture, and even labeling can influence how the brain interprets flavor. If you expect something to taste a certain way, that expectation can modify the sensory experience itself. This top-down influence interacts with the bottom-up signals from taste and smell, creating a dynamic system that determines how food is perceived and evaluated. Through this continuous interaction between taste and smell, the brain develops a detailed and flexible system for assessing what you consume. It not only identifies flavors, but also attaches meaning and value to them, guiding choices in a way that reflects both immediate sensory input and accumulated experience. The role of chemical cues in driving motivation and behavior is rooted in the brain's ability to use smell as an early warning and reward detection system. Odors in the environment carry information about food, danger, and social context, and the brain is wired to respond to these cues rapidly. What makes this system particularly influential is that it can activate motivational circuits without requiring conscious decision-making, meaning your behavior can be shaped before you are even aware of why. When you encounter a smell, the olfactory system sends signals to brain regions involved in reward and motivation, including the dopaminergic pathways. If the odor is associated with something beneficial, such as food or a positive past experience, it can trigger a release of dopamine, which increases the desire to approach and engage with that stimulus. This is why the smell of food can suddenly make you feel hungry, even if you were not previously thinking about eating. The brain is not just detecting the odor, it is predicting a rewarding outcome and preparing the body to act. This predictive aspect is critical. The brain uses past experiences to assign value to specific smells, and those values influence future behavior. If a particular scent has been repeatedly linked with a positive outcome, the brain strengthens that association, making it more likely that the same odor will trigger a strong motivational response in the future. This process is a form of associative learning, where sensory input becomes a cue for expected results. On the other side, unpleasant or harmful odors activate avoidance systems. Smells associated with spoiled food, toxins, or unsafe environments can trigger immediate aversive reactions. These responses often involve activation of the amygdala and other regions that process fear and threat. The body may respond with changes in breathing, heart rate, or even nausea, all designed to discourage interaction with the source of the odor. This rapid avoidance mechanism is essential for survival, as it helps prevent exposure to harmful substances. Chemical cues can also influence behavior in more subtle ways by altering mood and attention. Certain ambient scents, even when not consciously noticed, can affect how alert, relaxed, or focused you feel. This occurs because olfactory input interacts with systems that regulate arousal and emotional state. For example, some smells may promote a sense of calm, while others can increase vigilance or alertness. These shifts in internal state can then influence decision-making and performance on tasks. Another important aspect is that these effects are highly context-dependent. The same smell can produce different behavioral responses depending on the situation and the individual's prior experiences. A scent that is associated with comfort in one context might be neutral or even unpleasant in another. This flexibility allows the brain to adapt its responses based on changing environments and needs. Hormonal state also interacts with these processes. When the body is in a state of hunger, stress, or fatigue, sensitivity to certain smells can increase, and the motivational impact of those smells can become stronger. This ensures that behavior is aligned with current physiological demands, making the system more efficient in guiding actions that support survival. Because chemical cues operate continuously and often outside of awareness, they provide a constant stream of information that shapes behavior. They influence when you feel motivated to seek something out, when you choose to avoid it, and how you respond emotionally to your surroundings, all through fast and deeply embedded neural pathways. The influence of pheromones and body odors on social and reproductive behavior is a complex and often subtle aspect of human biology, but it plays a meaningful role in how individuals perceive and respond to one another. Unlike visual or auditory signals, which are consciously processed, chemical signals are often detected below the level of awareness, yet they can still shape attraction, mood, and interpersonal dynamics. In many animals, pheromones serve as a primary mode of communication, directly triggering specific behaviors such as mating, aggression, or territorial responses. These signals are typically processed through specialized pathways, including the vomeronasal organ, which is highly developed in many species. In humans, the existence and functionality of this organ are less clear, but there is strong evidence that chemical communication still occurs through the main olfactory system. Body odors carry information that the brain can detect and interpret, even if the individual is not consciously aware of it. One of the most studied aspects of this process is how body odor can signal genetic compatibility. Research suggests that people may be naturally drawn to the scent of individuals whose immune system genes differ from their own. This is thought to be linked to the major histocompatibility complex, or MHC, which plays a role in immune function. From an evolutionary perspective, selecting a partner with different MHC genes could increase the likelihood of producing offspring with a more robust immune system. While this process is not deterministic, it highlights how chemical cues can influence attraction in ways that go beyond conscious preference. Body odor also conveys information about emotional and physiological states. For example, when a person is stressed, their body releases different chemical compounds through sweat. These compounds can be detected by others and may influence their own emotional state, often increasing vigilance or tension. This form of chemical signaling acts as a kind of nonverbal communication, allowing individuals to share internal states without explicit interaction. It operates alongside facial expressions, tone of voice, and body language, contributing to a more complete understanding of social context. Hormonal fluctuations further modulate how these chemical signals are perceived. Sensitivity to certain odors can change depending on internal biological states, such as phases of the reproductive cycle. This means that the same individual may perceive another person's scent differently at different times, influencing attraction and social behavior in a dynamic way. These shifts are part of a broader system that aligns perception with physiological priorities. Another important aspect is how these signals interact with learned experiences and cultural factors. While there may be biological tendencies in how certain scents are perceived, personal history and environmental context can shape these responses. A particular scent might become associated with positive or negative experiences, altering how it influences behavior. This adds a layer of complexity where innate and learned factors combine to determine outcomes. The effects of pheromones and body odors are often not strong enough to dictate behavior on their own, but they can bias perception and decision-making in subtle ways. They may influence first impressions, feelings of comfort or discomfort, and the overall sense of connection between individuals. Because these processes occur largely outside of conscious awareness, they can shape interactions without being easily identified or explained. Through this continuous exchange of chemical information, the brain is able to gather additional data about the people in the environment. This information integrates with other sensory inputs to guide social behavior, contributing to patterns of attraction, communication, and emotional response in ways that are both biologically grounded and highly adaptive. Learning and memory through sensory associations is one of the most powerful ways that smell and taste shape behavior, because these senses are tightly linked to brain systems responsible for storing experiences and predicting outcomes. When you encounter a particular smell or taste, The brain does not process it in isolation. Instead, it rapidly connects that sensory input with the context in which it occurred, including whether the experience was positive, negative, or neutral. These associations are then stored and used to guide future behavior. This process is highly efficient because it is directly tied to survival. If a certain food provides energy and causes no harm, the brain marks that combination of taste and smell as rewarding. This increases the likelihood that you will seek it out again. On the other hand, if a food is followed by illness or discomfort, even if the cause is not entirely clear, the brain can form a strong aversion after a single exposure. This phenomenon, often referred to as conditioned taste aversion, demonstrates how quickly and effectively the brain can link sensory input with outcomes. The neural basis of this learning involves close interaction between the olfactory system, the gustatory system, and memory-related regions such as the hippocampus. At the same time, the amygdala assigns emotional value to the experience, determining whether it should be approached or avoided in the future. These systems work together to create a unified memory that includes not just the sensory details, but also the emotional and physiological responses associated with them. One important feature of this type of learning is its durability. Memories formed through smell and taste tend to be long-lasting and resistant to change. This is partly because they are encoded with strong emotional components, which enhances memory consolidation. As a result, certain preferences or aversions can persist for years, even if the original context is no longer relevant. This can be beneficial when it helps avoid harmful substances, but it can also limit dietary variety or influence behavior in ways that are not always adaptive. Repetition plays a key role in strengthening these associations. Each time a particular smell or taste is paired with a similar outcome, the neural connections involved become more robust. This process, known as reinforcement, makes the response more automatic and predictable over time. For example, repeatedly experiencing a pleasant taste in a positive setting can increase preference, while repeated negative experiences can deepen aversion. Context also influences how these associations are formed and expressed. The same taste or smell can be perceived differently depending on the environment, emotional state, or expectations. If a food is consumed in a stressful situation, the brain may link that sensory experience with discomfort, even if the food itself is not inherently unpleasant. Conversely, positive social settings can enhance the perceived value of certain flavors, strengthening preference through association. Another layer of complexity comes from the brain's ability to update these associations. While some memories are very stable, others can be modified through new experiences. Gradual exposure to previously disliked tastes, especially in a positive context, can weaken negative associations and create new, more favorable ones. This flexibility allows behavior to adapt over time, even though the initial learning may have been strong. Through this ongoing process of association, the brain uses smell and taste as tools for predicting outcomes and guiding decisions. These sensory experiences become embedded with meaning, influencing what you choose to approach or avoid based on patterns established through past interactions. Hormonal modulation of smell and taste sensitivity is a key mechanism through which the brain aligns perception with the body's internal needs. The sensory systems responsible for detecting odors and flavors are not fixed in their sensitivity. Instead, they are continuously adjusted based on physiological state. Hormones act as signals that inform the brain about conditions such as hunger, stress, energy balance, and reproductive status. And these signals directly influence how strongly certain smells and tastes are perceived. One of the clearest examples of this is the effect of hunger on sensory perception. When the body needs energy, hormones such as ghrelin increase, which can enhance sensitivity to food-related smells and tastes. As a result, foods may seem more appealing, and the motivational drive to seek them out becomes stronger. This is not just a psychological effect, it reflects a real change in how sensory input is processed. The brain essentially amplifies signals martubat with food to encourage behavior that restores energy balance. In contrast, after eating, hormones like leptin and insulin signal satiety and reduce the sensitivity to those same cues. Foods that seemed highly appealing when hungry may lose some of their attractiveness once energy needs are met. This shift helps prevent overconsumption by decreasing the reward value of additional intake. The dynamic adjustment of perception ensures that behavior remains closely tied to the body's current requirements. Stress hormones also play a significant role in modulating these sensory systems. Elevated cortisol levels can alter both smell and taste perception, sometimes enhancing sensitivity, and other times diminishing it, depending on the context and duration of the stress. Acute stress might heighten awareness of certain environmental cues, including odors, as part of a broader increase in vigilance. Chronic stress, however, can disrupt normal sensory processing, leading to changes in appetite, food preferences, and overall behavior. Reproductive hormones introduce another layer of modulation, particularly in how individuals perceive social and environmental cues. Fluctuations in hormones such as estrogen and progesterone can influence sensitivity to specific odors, including those related to other individuals. These changes can affect attraction, preference, and social interaction in subtle but measurable ways. The brain uses this hormonal information to adjust perception in a manner that is aligned with reproductive timing and priorities. At the neural level, these hormonal effects occur through interactions with both peripheral receptors and central processing areas. Hormones can influence the responsiveness of sensory receptors in the nose and tongue, as well as alter activity in brain regions that interpret these signals. This means that modulation happens at multiple stages, from detection to perception to behavioral response. Another important aspect is that these changes often occur without conscious awareness. You may not notice that a particular smell seems stronger or that a certain taste is more appealing than usual, but these shifts can still influence your decisions. Over time, this creates patterns of behavior that reflect the body's internal state, even if those patterns are not explicitly recognized. This system is highly adaptive because it ensures that perception is not static, but responsive to changing conditions. By adjusting sensitivity based on hormonal signals, the brain can prioritize the most relevant information in any given moment. This allows for more efficient decision-making, as attention and motivation are directed towards stimuli that are most likely to support survival and overall function. Hormonal modulation of smell and taste sensitivity is a key mechanism through which the brain aligns perception with the body's internal needs. The sensory systems responsible for detecting odors and flavors are not fixed in their sensitivity. Instead, they are continuously adjusted based on physiological state. Hormones act as signals that inform the brain about conditions such as hunger, stress, energy balance, and reproductive status. And these signals directly influence how strongly certain smells and tastes are perceived. One of the clearest examples of this is the effect of hunger on sensory perception. When the body needs energy, hormones such as ghrelin increase, which can enhance sensitivity to food-related smells and tastes. As a result, foods may seem more appealing, and the motivational drive to seek them out becomes stronger. This is not just a psychological effect. It reflects a real change in how sensory input is processed. The brain essentially amplifies signals related to food to encourage behavior that restores energy balance. In contrast, after eating, hormones like leptin and insulin signal satiety and reduce the sensitivity to those same cues. Foods that seemed highly appealing when hungry may lose some of their attractiveness once energy needs are met. This shift helps prevent overconsumption by decreasing the reward value of additional intake. The dynamic adjustment of perception ensures that behavior remains closely tied to the body's current requirements. Stress hormones also play a significant role in modulating these sensory systems. Elevated cortisol levels can alter both smell and taste perception, sometimes enhancing sensitivity, and other times diminishing it, depending on the context and duration of the stress. Acute stress might heighten awareness of certain environmental cues, including odors, as part of a broader increase in vigilance. Chronic stress, however, can disrupt normal sensory processing, leading to changes in appetite, food preferences, and overall behavior. Reproductive hormones introduce another layer of modulation, particularly in how individuals perceive social and environmental cues. Fluctuations in hormones such as estrogen and progesterone can influence sensitivity to specific odors, including those related to other individuals. These changes can affect attraction, preference, and social interaction in subtle but measurable ways. The brain uses this hormonal information to adjust perception in a manner that is aligned with reproductive timing and priorities. At the neural level, these hormonal effects occur through interactions with both peripheral receptors and central processing areas. Hormones can influence the responsiveness of sensory receptors in the nose and tongue, as well as alter activity in brain regions that interpret these signals. This means that modulation happens at multiple stages, from detection to perception to behavioral response. Another important aspect is that these changes often occur without conscious awareness. You may not notice that a particular smell seems stronger or that a certain taste is more appealing than usual, but these shifts can still influence your decisions. Over time, this creates patterns of behavior that reflect the body's internal state, even if those patterns are not explicitly recognized. This system is highly adaptive because it ensures that perception is not static, but responsive to changing conditions. By adjusting sensitivity based on hormonal signals, the brain can prioritize the most relevant information in any given moment. This allows for more efficient decision-making as attention and motivation are directed towards stimuli that are most likely to support survival and overall function. When you understand this interaction, it becomes clear that your sensory experience is not just about the external world, but also a reflection of your internal biology. The brain is constantly tuning perception based on hormonal signals, creating a dynamic loop where what you need shapes what you notice and what you notice shapes how you behave.